Let's go ahead and launch into this. Let me take just a couple of minutes and try to reiterate what I thought that we might talk about, and then we can actually change the topic to be whatever we collectively think is most productive. But one of the things that we've been struggling with in the I think Atlas project is how it is that one defines difference from normal. And if you think about the immune system, that's fairly straightforward because you're talking about components of a system that don't fundamentally change too much in their molecular state and composition as you go from a tumor state to a normal state. Organization isn't, well, actually organization is important, but when you get into the tumor world, one of the things that we lack is a definition of what normal is in terms of cell composition and in terms of cell organization. And I think one of the real challenges in the next generation of Atlas production is how it is that one specifies the ontology or taxonomy of organization. It's easy to think about how it is that you define cell types, but it's hard to think about how it is that one captures the concept of cell organization. And so I was hoping that we might get some discussion about ideas that might lead us in that direction. And we have cell organization and we have intracellular organization, the components that comprise the cells. And so those were the general topics that I'd sort of put on the table for discussion, but let me throw it open to comments at this point. I mean, there are certainly many ways that we can take this, but the idea was to try to get us to the point where we could compare tumor to normal and where we could also compare across tissues. So thoughts on those topics?
Guys, forgive me breaking in very quickly. I want to make sure that you've got a scribe in this group who can capture what's going on.
We do. Philip is
excellent. Brilliant stuff. Remember to take a break whenever is convenient to you, Joe. I don't want people to get fatigued and tired and overwhelmed. Let me know if you need anything.
Joe, can I ask a clarifying question? This is Zorina Gallus. I wanted to ask you if you mean in terms of organization, visually looking at tissues or having a way of referring to relationships or all of the above. I mean, we obviously know that pathologists can look at a tissue section and tell us if it's normal or not normal based on looking at organization of tissue within what they see.
I was more thinking about a quantitative definition of organization. I think that one of the concepts behind a spatial atlas is that the interactions, if you will, that result from changes in organization actually change the physiology of the cells that are doing the interaction. And so I think that what I was imagining is that there might be some way to define interactions. So an immune cell next to an epithelial cell, a vascular cell next to a fibroblast, et cetera, in ways that actually drive the physiology of the individual interacting cells. And just as an example, one of the crudest measures that we've started to play with in that area is the concept of entropy, how disorganized the cells actually are in the tissue. And so that's
so in that light, would it be interesting to develop patterns that are known to be normal for various tissues? Because the patterns would contain obviously relationships between cells, cell sizes, numbers, things like that.
Yes, I think that's exactly the kind of thing that we ought to be doing. And hadn't thought about putting it in that context, but yeah. If we could come up with saying, with an interaction atlas of normal tissues, then you could ask, okay, how far does a diseased tissue deviate from that set of normal interactions? That'd be actually fairly easy to do, I would think.
Yeah, and I think you can use machine learning at this point in time to actually do that. As you know, HapMap is looking at normal tissues, so it should hopefully help with the idea of what's normal for various organs and compared to tumor state or disease state in the same organs.
So how, maybe you could elaborate a bit on what HubMap is doing there in terms of defining these interaction patterns. Is that something that is already underway in HubMap?
There are several people from HubMap I see as part of the group, but I don't think that that's a specific topic that we are looking at. It's not that we couldn't look. I think there are technologies now that can actually define by cell signatures what are the interactions. I think spatially talking, as I said, I think we can look at what's the normal pattern of various tissues. I don't know that we have specific initiatives. Ajay or Jay, tell me if I'm wrong.
I mean, I think it gets into the common coordinate framework a little bit, right? And there's a lot of effort on that in HubMap.
I think that there are, yes, there are efforts regarding that. I'm more familiar with the idea of using the vasculature as a way of looking at patterns, distances from various cells and things like that. But there are, you know, going across scales. Kati Borna developed a model of the kidney because we have to settle on something. Rahul Satija is looking at the lung and how to define coordinates in the lung. So I think we have specific cases that we are looking at. But I'm not sure that we are looking at patterns. I've been trained as a pathologist, so I can tell you that the human eye can tell if the pattern is normal or abnormal. This is how we all get diagnosis anytime anybody is diseased, right? So if the human eye can do that, it means that the machine could do it even better.
Well, but I think that one of the opportunities here is to take it sort of a functional step forward, and that is to say, okay, if I've got a different kind of interaction, how does that change the physiology of the interacting cells in the context?
Correct. So if you get the signature, single cell signature from various cell types, and you can also see how they relate to each other spatially, then we can develop those kind of, you know, atlases based on interactions.
Well, since we have a fairly good representation from HubMap, maybe you could take just a few minutes and say what some sort of, what the general ontology development or taxonomy development activities are that are currently underway there, just so that we can think about building on those.
Anybody else wants to speak up? Am I the only one?
So this is Ajay. So the baseline ontologies that we are doing that's sort of relevant to what we are discussing are related to how we are adapting Oberon to represent hierarchical information about organizations within a tissue. And that's
Expand a little bit on that. Just say what you just said in a 5 times more words.
There isn't that much work yet in that level of detail. So what we are planning to do, so there are 2 sort of nascent efforts. The first one is basically trying to locate, and I guess some of the people in this group have already heard me say this yesterday, is actually locating where a sample of tissue comes from within the context of an entire organ represented on a computer. So that's one. So where, you know, the person slicing the tissue goes in and places it with reference to, you know, whether it was in the upper left quadrant, 3 millimeters away from some edge or some structure in the organ. So there's that sort of an effort. Then there is an effort. So Oberon. So I assume people know what Oberon is. Oberon is a taxonomy. It basically took all of the different anatomies, descriptions in different organisms and merged them together into that describe an organ and breaks it up into a set of nested hierarchies, so you can find out where a particular cell belongs or a cell type belongs or a substructure within an organ belongs. So it's a hierarchy because when you look at it as a human being, but at the back end, it's an ontology. So it has a part of definition, it has substructure definitions and other such ontological descriptions that help you relate and do computations on structures. What it doesn't have, and this is something I think in one of the breakout sessions yesterday we were discussing, what it doesn't have is any sort of spatial relationship yet. It doesn't, say this substructure within the organ is usually found at this distance from some other normal entity. So anyway, I have taken now more than 5 minutes. I'm not sure that I have explained anything much better.
No, that was quite helpful. So I don't know as much about the Oberon effort as at least I should. Who's running that? Or how is that being run?
So Oberon, as far as I understand, it doesn't actually really have a specific grant associated with it, but it's a bunch of different funding sources that they have managed to put together. So it's Chris Mungle, Peter Robinson, and Melissa Handel, who
I think they have funding, say, for the kidney from the Kidney Precision Medicine, right? So they are trying to engage domain specialists because it's an ongoing effort. I mean, if you look for any specific organ, you may not find a lot. It's ongoing, and they are looking to recruit both people who can help them develop the ontologies and who can get funding to do that. So
I could, sorry.
Go ahead.
Yeah, David Asumi here. So I worked a little bit on Oberon, and I work quite closely with Chris Mungle. So yeah, its origins are in pulling together a lot of different anatomy ontologies. It's multi-species, which is good for some things, but then if you're trying to build very human-specific views, then the labels are not necessarily the best. But we do actually have ways to adapt it. For example, using the human-specific labels coming from the foundational model of anatomy, which is very old and quite potentially useful, but very large and ungainly ontology of human anatomy. So I think, the challenge now is we've got this quite complex structure, which is quite rich. As AJ gave a really nice introduction there, it doesn't include spatial information, and that could be really useful in some sense, although maybe that needs to be relative spatial information. We do have the ability to do a certain amount of spatial reasoning in these ontologies as well, kind of sort of very basic deductive reasoning. But that's part of how these work, and there's potential to expand there. And there are links already, quite extensive links into the cell ontology. So there's a bunch of sort of usable infrastructure there, I think, but it requires updating, review, and some work to kind of provide sort of simplified views that restrict what's shown for the use cases, for different specific use cases. Like for specific, you might want a particular view that's relevant to human kidney, work on the human kidney or work on human lung, for example. Yeah, and I think so. Funding has tended to it's used extensively by the gene ontology as well. So there's a lot of there's a certain amount of work that's gone into it from there, and the various different phenotype ontologies including the human phenotype ontology. So someone mentioned Peter Robinson's work there. So I think it provides a good basis to build on, but there's definitely, and as I understand it, there's some interesting work going on with the kidney group in the HCA or at HubMap to kind of adapt it to make it more directly useful to the project. And I'd be really interested to understand more about what's going on there. Okay, I'll stop talking. Hopefully that's given you a little bit more background.
And I'll say one more data point if it is relevant. I think it's not money at this point for our efforts that is stopping development of Oberon or similar things or adapting Oberon, or it's actually people with enough know-how to be able to do things and to find them and find time on there. There are very few people who can actually do this, and that's my understanding of the world, is that's where we are stuck.
So one possible action item that we might take on is how to promote the further development of the Oberon community by joining in it and bringing in some of the people from the various communities that we're in.
Yeah, I think that would be fantastic. It's always the best parts of Oberon are just like the best parts of Go or any of these ontologies are cases where there's been really good community engagement. There was quite extensive work on neural crest and on facial musculature and things like that. So there's a few cases where there's been really good expert input, and the quality, inevitably is higher. I should say that there has been some contact with to try to tap the expertise within Oberon and the Obo community, but I think it's been it just needs kind of a longer timeline so that we can move resources around to do work. But so some of it's kind of, rather than coming in through grants, it's coming through sort of informal approaches.
Another potential is to have sort of jamborees or whatever you want, the efforts, everybody gets on and try to work together. I was part of I mean, at least I was a spectator. I also participate with what I know about the kidney. So I think it was a pretty useful exercise, because in a couple of hours, they got a lot of people in the same room thinking about the same thing. So they achieved quite a bit. So I don't know if that's another type of effort that could be done across various atlases.
So Philip, let's pick that up as a potential action item here, as something that we could recommend as standing up a few. What I'm hearing is organ site specific jamborees that might build on the Oberon concept and ask the question to how, I could easily imagine having the kidney folks tell us what they've done and then ask, let's say, a breast group to ask how well that would apply to a different organ, something like that. But that's the sort of thing that I think, from a HTAN perspective, would be something really important for the early cancer programs to take on, since that's where you're seeing the smallest changes away from normal.
Also, a question that I think we could also bring up is, things like Oberon, it's more, normal tissue, kind of the architecture of that. So there's kind of that scale at the, within the organ tissue level, but then also at the, cell-cell interaction level, like we were talking earlier. And those are two kind of very different things, but they do integrate at that, interface of scale. But, I think those are two kind of distinct domains, maybe. And then also, when we're doing these definitions, I mean, spatial component is one, but if you're looking at the cell-cell interaction side of things, how are you defining that in terms of the modalities you're looking at, right? So if you're looking at, immunofluorescence-based analyses, that's going to be very different than spatial transcriptomics or things like that. So that's another consideration that I think is going to be well suited to cross-consortia type collaboration work.
Yeah, I think that That's a really important point, how one defines a set of cell types depends on the tools that you use to define them. And there's probably a continuum of cell types if you deploy all of the different tools that we have. So I think one of the things that the community is going to have to figure out is what constitutes a cell type. You know, how are we going to arrive at a consensus of that? And how many any ideas, Andy, about how to do that?
So this came up yesterday in the multimodal tools section, where you know, we'll often see in single cell data where you have single cell ATAC-seq has, you know, splits this into several cell types, where single cell RNA-seq it's all glubbed together, or vice versa, right? So depending on the modality, you'll get different numbers of subdivisions. And that's even true if you're getting ATAC and RNA from the same cells. You might have, you know, everything clumped together if you're just looking at ATAC versus splitting out if you're looking at RNA. So it's definitely modality dependent. And I see Amy saying there's a blog about the topic. I think it's something that's come up quite a bit. And then also, depending on where you draw the line, you know, you can have, you know, there are papers that show, you know, tons of different cell types, and then some that just glob them together, and there's a lot less. So, you know, kind of having a hierarchy about that, I think makes sense.
Yeah, I can just quickly weigh in on the cell types for the brain. It's not just what defines a cell type, but one of the things we were trying to do for brain atlas work was make sure that while not defining the cell types solely by transcriptomic properties, that we were anchoring rubric around how we would do that, because the transcriptomic profile is one of the only ones that can be done across all organ systems. So, you know, you can't have, for example, meaningful cell morphology necessarily for all kinds of blood cells, but you know you're going to get some sort of transcriptomic value off any cell. So we're thinking of sort of the idea of the transcriptomic one being sort of this Rosetta Stone across all organ systems, and then a second order set of features would be related to cell specification based on that organ system. For brain, it's electrophysiology, things like that. But one of the things that went with this that I just posted to the chat section was we tried to also define a quantitative way of grouping and then creating taxonomic patterns or definitions around that. So this is just one proposal. We tried to actually create it as an option for getting weigh-in from the community. We had a whole workshop around this. And there's now subsequently, I think, a number of different review articles that multiple different people are preparing with multiple different views on this. So yeah, as Marianne says, it's lumpers and splitters. There's never going to be one answer, but I think what helps is providing mechanisms. So that's what we've learned, is when you have a mechanism on how to group cells or how to analyze your data where you can extract out this information, those are things that are going to move people forward rather than just kind of soapboxing it. So we do both, but the reason why I pasted that in is just because it provides a bit of a working definition of how you could use a particular approach to classify and assign taxonomy groups to cell types that we apply for brain, but should be extensible beyond that. Just one approach.
So to that point, how do you imagine that the community is actually going to converge on this? Are we just going to have a whole bunch of different strategies and we use them all, or?
I'm looking at Marianne because she has the best narrative on this of like, just abandon hope. There's not going to be a sudden kumbaya moment of community alignment, but I think that what'll happen is convergence of certain methods. So we already do this. Things like UMAPs, for example, people are using all the time to show aggregates of cell groups, and then formalizing that at increasing levels. And you know, again, getting applied procedures that work, that help people understand their data, will be more adopted than just somebody saying, use my term. But the thing that we're finding is the levels and the thresholds for what is useful are really different based on the different origin tissue types. So some of the things work well for one organ system and less well for another in terms of isolating marker genes. In a different session we were talking about maybe using something like a particular nuclear marker, say from a perivascular cell to a vessel, because you might be able to find that kind of marker in other sorts of data types, tissue types. But I don't think there's going to be alignment. I think there's going to be differences, but needing to sort of mosaicize or overlap different approaches across different organ systems. That's, again, that's just what, that's not from experience, that's from intuition. But from brain, we've been trying to also then apply a procedure by which you would make decisions to classify. And yeah, Marianne has had a lot more experience than I have just in terms of the community component of finding alignment, which is difficult.
Why we pretty much give up. No, sorry, I wasn't in the first part of this. I was in another session, but I mean, it's lumpers and splitters. We're still arguing and we're always going to argue. And I think trying to get these discussions on a more computational footing. So one of the things that we try to promote is if you publish these classifications and things buried inside of PDFs and have to do a lot of text mining to extract them, it's very difficult to come to any consensus. And I think, the more we use more standard terms to at least describe phenotypes and use terms, that are in common across from ontologies like the cell ontology, attached to data though. It has to be attached to data because you have to be able to, you have to be able to compare across these in some ways, and just giving a categorical label without any backup, it's very difficult to know whether you mean the same thing. But I think the exciting thing that is happening is starting to learn how to communicate in standardized, ontological terms. So at least we don't have to deal with all of the terminology problems, but attached to these pools of data that let you go in and independently verify. And then whether you think you have a reason to group it this way and someone else thinks they have a reason to group it that way, those are the arguments that go on. But at least we have access to the underlying data and at least a common vocabulary as much as possible for describing phenomena. That's sort of the approach that I think will work the best. And I know David and others have been working on things like that.
So what I'm hearing so far is that these ontologies are being defined pretty much by organ site and functionally, or at least ideally they're being defined functionally. They're functionally useful. So there's a lot going on out there. I think that we are probably at our break time. One of the things that I recognize based on the discussions that we just had here is that there are a lot of different programs that are out there, H10 included, that are attempting to solve the same problem, which is to define the spatial ontologies, not just spatial, but sort of cellular and spatial ontologies that we're all going to have to interact with. I think it's true that they're likely to be somewhat organ specific, but I think the general principles are likely to apply across organ types. And so I think the suggestion was made to think about a jamboree, sort of a spatial ontology jamboree, which would be populated by participants from as many as possible of the different organizations that Sarah highlighted in the introduction. Some of them will be well along and they could present case studies, and the others of them, H10 included, are pretty nascent in their efforts to try to come to grips with this, and they could be more in the spirit of trying to learn from what people have already done. And so I think that the proposal to establish some sort of a jamboree, again probably led by the people who are the farthest along in this, to bring everybody else on board, would be very helpful. So I'll just throw that out there as one topic for something that we might suggest. Any comments on that?
Joe, I'd like to ask a question to the room. It's more of a computational question. That is, when we get more and more images to describe spatial structure, and then we'll coalesce toward the description of the norm for a certain organ on all scales, on the whole organ, then smaller and smaller down to single cell. How do we measure kind of the distance from the norm, or eventually capture the range of variation among the healthy around that norm? Because if we don't have metrics to measure distance, there isn't an easy way to know whether we've captured the norm, or whether the first 10 samples is enough, or whether we need the next 100 samples. And then related to that is, how do we do this multi-scale integration? Coarser structure somehow is linkable to the finer structure. That would be my way of stating the question for now. I'd love to hear what progress has been made.
So, yeah, you're right. What we need are computational formalisms, not visual formalisms, that allow us to define those spatial relationships, and that can go across scales, and also that are somehow functionally motivated. And then there's the issue of what constitutes distance. There's immediate proximity, there is within the neighborhood, there is across town, etc. All of these things are functionally important at one level or another. And so I think that as we start going in the direction of developing computational formalisms to define that, we will develop the tools that we need. And I think the Allen Institute has been working on that for the brain. I think that there are probably a lot of examples that we could steal from the
I think this was discussed also in a session yesterday. It was a mathematical and computational session. The specific idea of the cell relationships and computational tools.
So I guess what we're getting to is the concept of data-driven quantitative ontologies, which include both the cell type and the cell spatial relationships. And so, Philip, in the spirit of actually proposing something down in the blue section of the document, I would propose maybe the development of a jamboree that would focus specifically on that topic.
I agree. We should probably define the discussion points, right? Like, I don't know if what we've just gone over would be all part of that jamboree, or like.
If you just took the sentence that you just typed,
yeah,
just that one right there, and put that down in the blue box.
I have trouble with the formatting in this Google Doc, but all right. Okay, so this would be the recommendation, right? So that the problem then.
Well, the problem is to develop data-driven quantitative ontologies that include cell type and spatial information.
Well, I think that's, we had some discussion of that in the cell ontology meeting yesterday in the breakout. And there's some sort of preliminary work on that with the Allen as well. But it's kind of a challenge in a research project. I think it's very necessary. But I wonder whether there's also a need for sort of simpler, more concrete kind of getting together and having some jamborees or working groups that work together with Oberon just to make it fit for purpose and to get the views and slices of Oberon that people need to use. So there's sort of some more mundane work that I think we could easily do. And then there's more kind of research project, we don't quite know how to do it yet work, which I think is the data-driven quantitative ontology work. So we do, the one case where I do have some experience in potential data-driven ontology work is with neurons in Drosophila, where we've been classifying by 3D morphology, and we get a clustering and a hierarchical classification that comes out of that. And we're looking at how we leverage that for building the classification into the ontology. That's a nice, relatively simple case.
Yeah.
Still hard, yeah.
Yeah,
And another thing could be just what are, for the different data modalities, what are the minimal kind of sets of, what is minimal data you could link to so that somebody, you know, could use that data in combination with their own data to try to automatically map onto. So you declare that you've discovered a new cell type. What's, for any given modality, what's kind of minimal data that you must register so that somebody else could potentially use that to map their own data onto it?
And I think broadly too, the ability to bridge between different data modalities. So, you know, if you're doing single cell RNA-seq, you're not going to get morphology, but you will get, you know, certain marker genes that could then translate to specific markers that you could look at the protein level and immunofluorescence, or so kind of what are the most valuable, I guess, bridging properties between modalities or between different assays?
So more as background information, the KPMP group held a wonderful, one of the best sort of ontology meetings that I've ever been to. And I think if we can replicate that to other organ systems, it would be a good start. The second thing I want to, and I feel like I'm going to channel Zorina here, the other lack of people that we are going to have are actual pathologists who are interested in this kind of stuff. It's not going to be just the technology developers doing RNA-seq who are needed, but that's another group we need to find. And
so I think Sarah Teichmann's been doing some work with pathologists just to map out classical, the standard markers that are used in the sort of classical or medical knowledge of cell types from pathology that are used diagnostically.
Yeah, so this is Zorina. So Ajay second the thing I said about the kidney jamboree. I think that's one thing that we already thought maybe we should propose for various organs or cell types or whoever feels like they know enough to help. But yeah, it's essential to have anatomists and pathologists because these are people who have learned about this and looked at this for ages, and we always get diagnosis based on what they see and what they know. So we should be able to at least start from the level of current knowledge and then make it better. The other thing I was going to say is that I'm aware of several papers, and I gave an example in the chat box, where single cell analysis data was used to actually infer spatial relationships between endothelial cells. As you probably know, they are a continuous surface that covers all the blood vessels. So people were able to show how gene expression varies as the endothelial cells move from the venous side to the arterial side. So you can actually infer relationships between, you know, where the cells are and ontologies based on single cell analysis. So that's a specific case, but I'm sure that similar things can be developed for other cell types and organs and things like that. So that was from the brain vasculature, and maybe some of you already know about it.
Is there a journal out there that spatial biology? I'm thinking of genome biology as sort of
I just got an invite 2 days ago about patterns or something like that, patterns journal.
If that mean, yeah, if you could send around information about that, it would be useful to include in this kind of a jamboree. It would seem to me that we're in need of some common place where people will begin to publish the ideas that are coming out in this field and help us to coalesce around some of the good ones.
This is Marianne. I think one of the things we're finding in the SPARC consortium, which I don't think is part of that, but that's the peripheral nervous system to end organ mapping. So we've been working with Oberon, and we find that a lot, you know, that the core ontologies provide you the concepts, but when you're actually trying to apply it to experimental data, they tend to get very precise, and there's cross products that you really want to be able to form. Because people are looking at, well, this is the anterior end of this organ, or it's a junction or an intersection. So I think in this ontology jamboree, making sure that we spend some time on what best practices are for how you use these base ontologies to form more complex structures, because there's several ways that you could do it. If one does kind of come to agreements about this, and my experience is most people just want a pattern that they can follow. They don't really, unless you're a knowledge engineer, have any desire to invent their own, that you could get a lot of traction in these types of jamborees if you sit not just on, oh, well, you're missing this structure or this structure, but when I apply it to experimental data, this is the model I have to use. These are the things that I have to pay attention to. And so I think organizing these meetings as a series that might occur, you know, on some basis and some overarching structure. There may be organ-specific ones, but then the groups may come back together to sort of share insights. I think that would be very effective rather than doing it in a one-off, because by the time everybody figures out what everyone else is doing, right, there's usually not all that much time to digest and think about it. So I think thinking about it as a series of meetings that allow us to spread out and come back together again might be really interesting.
So in your community, do you have things like challenges where people take a common data set and by different classifications?
No, we haven't gotten to that point yet. We haven't gotten to that point. The people who are contributing data in SPARC tend not to be kind of, I think SPARC is a desire to meld that into a richer community than it is, and then we can move on to these types of challenges. But right now it's just trying to get them in the tools that they use. So that's the other thing that we're doing in SPARC, is we're trying to get these ontologies in vocabularies into the actual tool set that the researchers use when they annotate their data. So they mostly use micro bright field tissue mapper. And so we've been working to engineer that. And then we have an anatomical working group that provides advice, because you see the types of terms that come in. We review each term to determine whether there is one already existing in an ontology. If they couldn't find it, if it's even a legitimate anatomical terms, we do find errors, right? We find errors that people say it's this, but when you probe them, it's actually something else. So it's been an interesting process on practical application of, it's mostly Oberon. And then if there are terms that aren't in Oberon, we've been gathering them up and we're going to submit them back to Oberon, because SPARC's main area of expertise is the peripheral nervous system, and none of these anatomical ontologies can be equally rich in all areas. They do need contributions from the community. So getting those pipelines and how we would do that, I think across consortia would be a really powerful organizational strategy, I think.
Pardon my ignorance, but what does SPARC stand for?
Stimulating peripheral activity to relieve conditions. So it is all, it's really geared towards bioelectric medicine. So there's a lot of work going on of stimulating nerves in the peripheral nervous system to affect, the central nervous system and things like pain. And so that's what it's called. It's terrible, terrible name.
I got it now. Well, that sounds like a really interesting series of meetings, that you could start off with a few case studies, and then you educate people who are trying to bring up new ontologies and new organ sites to solve new different problems, and they could work off of what people had already learned how to do. And the concept of bringing in some of the lessons from the Drosophila field is actually quite interesting. I mean, I think that's probably the most well annotated, spatially defined system around. So
the Allen Brain Atlas is pretty cool as well.
Okay.
One thing that I think is worth mentioning here is that we also have yesterday there was the meeting with the CCF, the Common Coordinate Framework, And that is basically a large discussion about the spatial relationship between, or how to describe the organ piece. And I think probably within that, I mean, that what we want to get into is diving deep into the interactions from there, and what types of cellular interactions are going on at a deeper level within the different structures that are defined in the CCF. So, I mean, it could be that this is a more in-depth version of what they're trying to do there, and I could see a collaboration there also, and that's not cross consortium necessarily, but it's within this group and the same sets of ideas. So I think that it could make sense for us to integrate ourselves with those ideas also, and connect with Katie Borner on this.
Philip, can you capture that?
Yeah, I just think that there's a lot of relationship between the types of things that we're trying to do here and what they're trying to do there.
Yeah, no, I agree with that. I'm just sitting here thinking about the HCAN project and the fact that we're just beginning to get started in this business, and there's already a lot of work that's been done that it seems to me that we should start with, not reinvent. And so this kind of a jamboree would be quite effective, or quite helpful, I think, in guiding our thinking.
Sure.
So Philip has been capturing this concept of a jamboree, or series of jamborees, in the blue template here. Can everybody take a look at that and see if we could perhaps refine what's up there?
Need some work in streamlining, I guess.
So I guess the title of the collaboration would be a collaboration to define or to develop data-driven quantitative ontologies. So I guess that first, yeah, to define, and then pick up the second sentence down there below. Good.
Would this also include an effort to figure out what are the technologies, the current technologies that allow us to do such correlations?
Yeah, let's capture that idea. That's right. Obviously, how these things are defined, it depends on the technology that you use to measure the system. So there could be an epigenomic ontology, and there could be a transcriptomic ontology, and a proteomic ontology.
Yeah, so I think it would be important to know that, because then you can compare or synergize or find ways of putting together the data that come from different channels.
And also, I think this is where, like that we were talking about earlier with the method for defining this. So, having kind of a uniform framework for that, because across all these different modalities, you might have, pretty large differences at the more granular level, but at the broad classification level, usually they can all come together and unify. There's a broad cell type classification is going to be identified in RNA and ATAC and cyclic IF, whether it's broadly immune cell population. So it kind of depends on what level of the hierarchy you're at to be able to bring those and unify across modalities. So I think that's
yeah. The other thing I think would be very helpful is to come up with some challenges or cases where we know about relationship between cells, let's say an immune cell in the normal context, out of context, or interacting with this cell A or cell B or cell C, and they take several modalities and say, show me how your modality can identify the neighbors of this immune cell. I don't know, it makes sense what I'm saying? I don't know. The other example I'm very familiar with is, again, going back to blood vessels. You have the endothelium that covers every blood vessel, but they go from super small blood vessels where endothelial cells have no other immediate neighboring cells, to large blood vessels where they have neighboring smooth mast cells and pericytes and all kinds of things. So in the signature of the endothelial cell, I should be able to read who is the neighbor. Or the endothelial cell is now in the kidney, so the neighbor will be a kidney cell. Can you show me this in the signature of this one endothelial cell? So examples like that where we have known associations and try to see how we can see those signatures through current methodologies, or how we can recapitulate things that we know.
Yeah, I think that'll be particularly valuable too when trying to describe the difference between normal and a disease state like in a cancer, because those vectors or signatures might be off in some way that kind of can capture some of that disorganization.
Yeah, correct. So I remember I used to be in academia myself, and we showed that the pattern of the small blood vessels that develop in response to tumor or hypoxia, whatever, is very different than the normal pattern of capillaries that exist in that tissue. So the signature was in the shape and size of the blood vessels, and it can be read. And I think some of you who look at tumors know that. You don't have blood vessels usually.
Cancer-associated fibroblasts, and there's a lot of different changes in normal cells within a tumor that are altered in their context.
And immune cells and things like that, right? So you can say, is this an immune cell sitting next to a cancer cell, or is this a immune cell sitting in its normal location in a lymph node or something?
I think to that point, we'd also have to define how well the cell type is actually defined and come up with metrics for that. So the likelihood that this thing is actually what we think it is. And also, where it sits on the spectrum of this cell type. So those sorts of classifications are also going to be important for people to see as well. Because even in the paper that you shared, Zarina, there's two different cell types I think they were talking about, and one of them, does pretty well on a continuum. The other one is much more binary looking, where it's like either this or that. But the other one is much. It's
correct. So cells that are going to be part of the functional units of various organs are very specialized. So there will be more kind of binary, and then there are the ones who are part of the continuum that goes from veins to arteries that are similar. Yet in there, they were able to show how the gene expression varies and where they were located based on their gene expression.
Yeah, and I think that also goes double for cancer, where the definition is hardly binary, and often you'll have signatures of a certain cell type that are unique to a patient just because of, and not even in the tumor, just in normal, the stromal cells, and how they've been altered in a unique way in that individual patient, and how we can, identify those, relate them to one another in some common framework.
Does that even lend itself to a common framework, or I'm still trying to understand how useful these ontologies will be in a advanced cancer context?
I think if you have a method, a hierarchy kind of infrastructure where you can say, well, yes, this is fibroblast, right, and it's altered in this way, so therefore it's likely cancer-associated fibroblast. Now you don't have to necessarily say that that's the same calf as this other one, right? But in a different patient, but just saying that at least having some framework for saying it is a cancer-associated fibroblast based on the hierarchy of cell type classification of the sample.
But you could also say this fibroblast should not be here, because in the normal tissue, you wouldn't have a fibroblast here, or you wouldn't have a fibroblast that has this marker, correct? So when you have a tumor, you have all kinds of cells that you normally are not there. This is what they see in biopsies, right? they see cells that were not supposed to be there. So that's an indication there's something wrong going on. And then you go on and see what type of cell, what characteristics, therefore maybe this type of cancer.
So one of the, I mean, sort of along these lines, one of the issues that we're trying to deal with in the cancer world is, does a cancer cell sitting next to an immune cell have the same pathway usage as a cancer cell sitting next to a fibroblast? There, you know, if it turns out that that's true, and we have the ability to actually quantify that using the kind of tools that we're talking about developing, then what you have is a mechanism of therapeutic response heterogeneity that we need to learn how to manage. And so it moves us in the direction of being able to think about that quantitatively.
Yeah, I mean, I think in some ways captured in the hierarchy, that would be, well, this is a cancer cell. And then within that next level of hierarchy, it would be, well, this one's next to an immune cell.
Right, right. Of course, you end up with 10,000 different cell types when you do that, but that's all right.
Yeah, but then you can go up a level, right? I think that's, you know, like just back to the fibroblasts, you know, in normal tissue, you're going to have fibroblasts. And, you know, if you want to do that connection between tumor and normal, you can say, well, they both fall under the broad category of fibroblasts. Now, then when you go to the next level of granularity and the hierarchical framework, then those differences start to come out.
So do you think you think you can achieve that using combination of single cell sequencing modalities and I mean, I'm trying to think more practical in terms of, if we have those jamborees, is there like a way to develop these hierarchies based on data that are already out there, or is it discussion based at this point?
I would argue that at the top levels, yes, because those hierarchies are going to be the same no matter what modality you look at, and they should be defined as such, right? Where the top level, you know, of the hierarchy is something that can be defined across all platforms, right? So very basic, you know, the most simplistic initial splitting of these hierarchies, and then, you know, down a level, you know, then it gets more into the modality specific type facets that are always going to be there. But if you move up to a level where, you know, let's say if you're doing, you know, genomics strategies, RNA and attack or whatever, yeah, you can identify this modality based on those, but maybe not with a certain morphology analysis, right? So that's where those might split up. But if you're just comparing between attack and RNA, then, you know, those, you could go to that lower level of the hierarchy and do your comparisons there. But at the top level, I think, you know, it has to be defined as something where it can be identified across all modalities and platforms.
But I think, Philip, we do, in fact, you know, let's say a year from now in the H10 project, we will have the data sets that I think could be used to begin to do this. And if we can start this by trying to define the ontologies up front, I think it'll probably color a little bit how it is that we collect the data to populate the ontologies. And rather than just measuring a bunch of stuff and hoping that it assembles into something useful later.
That's what we do, right? So what is your, Joe, you and the others, what's your vision for these jamborees? How do you see them work, and what do you want to get out of it? I mean, I'm just jotting down all the talking points and questions, and probably be good to have contact points.
So one type that I propose is that you get the organ specific or cell specific people together and put together the best ontologies that we are able to. And I can tell you that from looking at what they did in the kidney jamboree, it's kind of fun to watch, because even people who didn't want to participate were so upset about what other people would put in that they would have to go in. And, you know, it's, you know how scientists are, they may or may not want to participate, but
there's a sweet spot definitely for this, which is something which is good enough to be taken seriously, but bad enough to get people motivated to fix it.
Yeah, correct. So that's one that would be immensely positive, I think, for going across atlases and everything. I mean, if you have 15 or 11 lungs and I don't know how many kidney atlases, do they all use the same ontology? Do they all refer to the same thing? Or are we all talking about the same thing using different words? Or
so I think that might be the danger of having it organ specific, right? Again, people may talk in parallel rather than getting to that hierarchy that
no, because what you do is you have an organ specific specialist getting together and sorting out the organs, and then you have sort of a super group that looks at how to integrate across organs or how to put it back together in the body.
I mean, I think we have a certain amount of this already at a higher level in things like Oberon, where we have general terms for sort of gross tissue types, epithelia, stromal tissue, etc., that get applied across lots of different tissues, just like we have many cell types that get applied across lots of different tissues. So I feel like there's sort of two things here, and both of them are useful and important. One is to sort of get together and define a basic framework of which actually reflects a lot of quite old and basic common understanding of how to structure things, and making sure that it's up to date enough and that we've got views that people can use. And then there's the more cutting edge, how do we bring in data to define these terms as well. And I think the second one is, you know, they're both important, but the second one is harder. So I feel like I wouldn't want to, I'd be a bit worried about mixing up the two so much that we don't make progress in the first one.
See, I'm probably a lumper, because I was thinking that they are not separate. So let's say you have an ontology for epithelia, and then you ask people to contribute single cell analysis data to show how these epithelia, or prove that they are different depending on which organ you look or what neighboring cells they have. So you can split off and say, okay, we think that there are 5 types of epithelia, and now you show me that there are 5, or you maybe 10. And then we have to define new cells, or we have to define states, or so forth. So I think that it can all be connected. And maybe.
Yeah, I mean, some of it is sort of a matter of how specific it is. The higher level stuff can probably be done without that, but then at some point you get down to novel observations, and there, you know, we're going to need to be linking to data. One part of it can be a simpler way. One simple way to do it is to use links to data as evidence for structure in the ontology. A more, you know, a radical way to do it is to actually use data itself to structure the ontology, but then the question becomes which data, which that becomes difficult, you know. So I think with something like the Allen, there's such a large amount of work going on into the cell typing there that there's it's possible to come up with a standard based on, you know, this is all the stuff that's been integrated by the Allen right now, and that will be reasonably stable. It's harder to actually come up with sort of exemplar data for cell types or tissues from lots of different groups, but you can, you know, you can annotate the ontology with evidence taken and point that to the different data sets. That might be a more straightforward and rapid way we could start to integrate the data. Does that make sense to you?
Would you mind rephrasing that in one sentence as sort of a
Sorry, okay.
challenge?
All right. So so we could, okay. So the first bit I put on here is just the making sure that the general ontologies are fit for purpose and getting feedback. And I think that's a really quite constrained thing that we could have some really solid outputs from quickly. And then the next is the challenge of incorporating data. I think there's 2 ways to incorporate data. One is directly data-driven structuring the ontology. Another is using data as evidence to back up the ontology structure that we build.
No, you could see this also iteratively. I'm sorry. You can go back and forth between the 2, right?
Yes, I have a method I'd like to share with everybody, if it's possible. That kind of might solve what you're discussing right now. Is there any way I can like share like a screen or share like a PowerPoint slide?
There should be a button at the bottom of your screen that says share.
Yeah.
It's a green
Drag-
folder.
I see it. No, I see it. All right, so can y'all see this?
Not yet.
I wonder if the host has to be able to grant permission. Let's
You
see.
have to go on
Oh,
and,
you have to
click
select
on your,
what you're going to share. Yeah.
you have to click on the screen you want to share. So once you,
Oh, okay. Yeah, all right, let me see. Yeah. All right. Is it showing up now?
Yep.
Yep, there it is.
Okay, so I'm messing up now.
Yeah, we can see it.
All right. Okay, so here's the strategy I'm thinking about. So in the bottom, if I can get this out of the way. Okay, here you can see over here a section of a blood vessel. It's not quite the capillary level, but pretty close. And you see it's layered. It's got 3 layers to it. And I can get this out the way. Okay, at the bottom here, you can see each of the layers now separated out. You can just moved apart for the sake of demonstrating this process. If you see along the layer, you see these point locations. They're varied based on the spacing between these points. And the points are in these tables. These are the coordinates for each of the points based on which one of these models we're talking about. And the model is indicated by the color of the columns in the table. And if you go to this table here, you see it's like a hash table that's used to put together like a scheme, in this case for, you know, some kind of microscopic nanoscale fabrication strategy that was designed for. And here you can see there's different cell types, different extracellular matrix materials, growth factors involved, signaling factors, and these, and you know, environmental conditions, flow rates. These are just, these could be anything that you put in the column and provide, as you see on the other side, you have like a alphanumeric code that represents in each column that's located. And you see the columns per se are in the left, you see a part of this, the color code. In this case, it's the CAE lab code. And each column is, so like for the red, you see 5 fibroblasts, that's the 5, the first 5. 4 is trophocollagen, that's the 4 in here. And so on and so on and so forth. And when you put all these together and put them in this code, the code generates a color that represents these alphanumeric codes here. So, and that, from that color, it's applied to the surface of that particular model. And this particular model will represent the location of a specific cell type. Then each point, in this case, I don't remember what the spacing is on this particular one, but the spacing can be whatever you want the spacing to be. It could be 5 microns, 10 nanometers, point location spacing. And so you can arrange the spacing so they're located on a specific endothelial cell or below. So the endothelial layer would be in the innermost, smallest layer. Then the, you know, here would be smooth muscle cells, and here could be certain connective tissue layer, and you can make as many layers as you want to represent whatever tissue structure you want.
Hey, Bill?
Yes.
This is a really good example, I think, of the kind of talk that we ought to have at one of these jamborees. It seems to me that, I mean, I don't think that we have time to walk through the whole model here, but this is exactly the sort of thing that I think that we would want to trot out as an example of something that we might all build on. you know, especially if the code was available and the formalism was available, that way we wouldn't have to invent it. We could just basically steal what you've already got and move on. So yeah, that's a perfect example of what I think that we should try to accomplish in these. We have about, according to my clock, 15 minutes left. If
maybe what's needed is a planning committee that meets to try to hash out this idea of what would be effective. There's a lot of ideas in here. We probably can't come to a conclusion today necessarily, but having a planning group get together.
That's an excellent idea. Yep, we should do that. I think if we have a, if you scroll down, Philip, there is a section down there that identifies people who want to think about organizing stuff like planning committees.
I put my name down.
I think one of the everybody has to sign. Everybody who was part of the group has to put their names in to begin with. I think that was.
But I think it'd be good to single out a few people that will actually initiate or help initiate these jamborees, or contact people that could present, such as what we just heard, right, from Bill. I think, is that the kind of information we're looking for to get those started? Existing platforms, how to use them and incorporate them, or?
For sure, I think that the people who are principally involved in organizing these things should be well along the road to having attempted work in this area. So at least you know
that includes the H10, right? Fantastic.
I'm happy to help in organizing this, but I think that you know, some of the HubMap and Allen and other Spark initiatives might people from there might be better positioned. So who volunteers?
I wouldn't mind presenting, but I don't think I could be part of putting together.
I am proposing that Andy and Marion would be great.
I like that.
I'm happy to help, yes.
Yeah. No, I'm happy to help as well. I think, you know,
and David can bring the stuff from Oberon, I think.
Right.
Yeah, I'd really like to be involved. And we already have some contacts into with some discussion with Catty about working together. It's the slowness has just been kind of trying to redeploy resources at short notice.
So perhaps we can get new resources.
Yes, that would be great. I mean, I think we've got lots of interest and goodwill for, you know, for training and outreach and getting jamborees. You know, the same kind of discussions are going on for the cell ontology, but it's just having enough people in place to run these things as well, and how we do that. It's probably the limiting factor right now.
Well, you know, I think that, you know, some of the ideas that have come up here, code sharing and data sharing and things like that, are the sort of thing that there should be, let's say, governmental and philanthropic support for specifically, you know, in this consortium concept. And so maybe one of the things that could be accomplished in the development of these jamborees is to make sure that we engage representatives from the various funding sources that might be able to provide additional resources to move this along.
Absolutely. I think in one of my last sessions, you should think of this in terms of short-term, mid-term, and long-term funding.
Right. I mean, I think you're inventing a new field here, and so it's going to take work and sustained funding.
Yeah, and I think some of the initial steps that we'll want to take is to kind of just see what infrastructure is in place already and what's out there, and then see if the concepts that we've brought up here can link in and potentially be, you know, added on to some of that instead of reinventing the wheel.
Yep. So one of the things we heard about earlier on is the idea that there are going to be some 2-minute talks that propose different ideas. It seems to me that this Jamboree concept is something that either Andy or Marianne or David should volunteer as a 2-minute concept. I'm waiting for wild enthusiasm on the part of the 3 people.
I think Andy was about to say yes.
Yes, I think so.
I saw it.
No, I was not about to say yes.
Yeah. I mean, in terms of just giving one that says, I mean, not to that we're trying to plan a jamboree that will deal with all these. I think that's not hard, and I don't mind doing it if you want.
Okay, I don't
David,
mind.
Oh, you want to do it, David? I'll give it to you.
Okay, thank you. Cheers. Maybe if we're doing this at the end of the day, yeah. So if I draft a short bit of text and then people can chip in on it.
It would have to be somewhat concrete, though, right?
Yeah.
You'd have to.
Yeah, exactly. I don't want to get up there and just waffle about this.
Yeah.
General idea. So I think
how to contact the different networks and how to get this.
Yeah.
Well, and also I think, one of the concerns is if we, give a 2-minute talk and then someone's like, yeah, we have a consortium that's doing that right now.
Well, that's okay. I mean,
I think that's what this is for, really.
Yeah. And
I think if there are more people who want to do the same thing, it just gets stronger. So maybe the 3 of you can collaborate in doing the 2-minute talk, and one will present. But I think you can all just come together to write a 2-minute talk.
Yeah, and the easiest possible thing would be for somebody to stand up and say, yes, we're organizing that next week. Here's how you sign up.
Because as has been pointed out, these different sessions. And so if
we're losing you, Marianne. Say that again. We lost you. Now we've lost you.
Yeah, I'll type.
We have you back.
Back.
The themes have been coming sessions. So we don't want 5 jamborees.
Right. So I think the idea is we want to put forth the idea that we need a jamboree of this type. We don't have to own it, but we certainly want to encourage its development and would be willing to participate in its organization. Okay, so Marianne, the 3 of you are going to conspire offline, come up with a proposal for a 2-minute sales pitch at the end of the day.
Okay.
This has been a very educational session for me, I have to say. There are a lot of things that are going on that I didn't know about already. And so I think having this jamboree would be beneficial to a lot of people. Stop us from reinventing wheels.
Did everybody put their name in the document? I don't see the document, so maybe they did already. Just make sure to capture.
Philip, if you can capture everybody, that would be good.
Everybody can write their name in.
All right, I'm going to put my name at the bottom of this. There. If everybody else follows my name, then we'll know who was there. Oh, I see. You've got them all down there. Okay, we're going to put it down there.
So is the goal still to have those organ site specific, or is the goal to propose it more generally?
I think organ specific meetings would actually maybe, perhaps an overarching one, but I think organ specific would be a lot more productive.
Also, I think we should probably have breaks where the different organ specific ones come and tell everybody else what they've been doing? Otherwise,
I agree.
That's a good idea. Yeah.
I would start with an introductory jamboree where we get the organ site specific people to come in and tell the rest of us who are somewhat earlier in the phase here what's already been done.
Yeah, and we could get an overview of the ontologies, but then I think to actually do productive work, we'd need to break out into organ-specific groups. Look at a bunch of specific ontology details or whatever, data that we can use for structuring, and then maybe come back together.
That's a great idea. Yes, I think that's exactly the right format.
It sounds like a monthly event then, or something with somewhat regular meetings.
I think maybe quarterly or something. You don't want to overwhelm everyone with staring at ontologies is non-trivial even for professionals.
Well, but the various groups that are out there, I'll just take H10 as an example. We have working groups. And we don't have an ontology working group, but we could have an ontology working group, probably should. And so I think many of these programs could learn from this and then take it offline and continue to iterate on it and then come back periodically to the larger group.
And I think maybe we should think about how we fit in some training here as well.
I have a quick question, if I get a chance. You talked about specific organs. What about something like a vascular tree system that kind of goes from one organ to the next? It really it really crosses all the different organs, and you get much of the same type of structure. Has anyone thought about, how would you address that when you come from one organ to the next?
I think my understanding is that's a lot of what the Common Coordinate Framework is doing.
Okay.
So bringing having the Common Coordinate Framework people represented at an initial meeting, I think would be really critical, and I guess your input as well there, given that that's your interest.
Okay, thank you.
Yeah, I think one of the things that we need to do, again, if we get the opportunity to have this 2-minute sales pitch, is to ask how many different groups are there out there that are already engaged in this. I mean, we've picked up a goodly number of them, but there may be others. And so it would be useful to try to cast as broad a net as we can.
Yeah, I think that's the most important thing at the start, just because I know that the Human Cell Atlas has a big, consortium for figuring out ontologies of cell types, and then the Allen Institute certainly does, and then the Common Coordinate Framework is a whole nother thing. So I mean, I think the real goal would be trying to bring those together, and then if we wanted to get something that's more specific to kind of what we were initially setting out as well, is to bring in then how do we characterize the difference in cancer, right? So trying to pull in some of those consortia and what they've already built, and then adding on this layer of, okay, can that structure that's being assembled actually support this other vector of information, which is deviation from normal?
I think that's.
I think we are talking about different scales. I'm sorry, right? I mean, from single cell to organs to human body, right?
But I think you need to set it apart from all these efforts that are, Common Coordinate Framework efforts are ongoing, right? And I think what Andy said, how cancer deviates from these structures, I think is somewhat unique. And I don't think that's been tackled excessively, if at all.
And even just ensuring that whatever structures that they're setting up for their coordinate frameworks or cell type annotations or whatever, their infrastructure is for defining these things, making sure that there's a way to actually incorporate that information. I think that's maybe a key point.
Yeah, we don't want to, call something a different ontology than what they're calling it. Like, we need to be kind of unified in our approach. And you're right. I mean, their scale is going to be much larger than the scale that we're trying to get to with the cell types. But they're going to have certain structures that are going to have certain cell types in it. So we do need to come at this, in a teamwork sort of fashion with them.
The other thing I want to ask is whether we make a point that this is going to be human, right? I mean, we are looking at human ontologies.
Well, probably for the most part, but not exclusively. we're getting to the end of this. It seems to me that what you've really defined here is the serious need for this planning committee. I mean, we've made good progress in this, but I think that, you know, if you guys who are pitching this would pitch the idea of this is our contribution to a larger planning committee that might get together and define what all of the different opportunities are, and then organize a series of these things that would be populated by people who will self-nominate or will join somebody else's group, whatever.
I just want to ask if we captured stuff from the chat box. Different people gave different links. So is that already in the document, or should we, each of us who send links, put them in the document? Is that useful?
I was trying to capture some of them. I know the chat will be also recorded. I think we can maybe export this as a file. I'll do that.
Okay, thank you.
Oh, but then I can't copy and paste. Does anyone know how to do this easily?
Yeah.
Okay.
Okay, I think unless somebody has a burning topic, I think that we are done with this session. And well done, I might say. Thank you.
Thank you, Joe.
Bye-bye.
And everyone.
All right, let's go back to
Thanks, everyone.
home.
So feel free to work on that blue box.
Yeah, if there are links that Philip hasn't captured, just type them in.
Yeah, I think Aaron is copying the chat, right?
Perfect.
Thank you.
Thank you all.